Dear students, today I am going to tell you about measurement of pH by pH meter. So this is the topic, measurement of pH. This is part 1, and now the introduction of pH first. What is pH? It is defined as negative log of hydrogen ion concentration, where H is the molar hydrogen ion concentration. H is denoted by this notation. Here H is given with positive sign, and this bracket is used to show the molar concentration of hydrogen. This is not specific for hydrogen. Molar concentration of any element or compound is shown within the brackets like this. Actually, pH must be defined in terms of thermodynamic activity of hydrogen ion and not as molar concentration. Activity depends on the concentration of other electrolytes present in the solution. However, in biological systems like our body, the total ionic strength is always sufficiently low low that the effect of electrolyte concentration on the pH is small enough to be ignored. हमारी बॉडी में जो बॉडी फ्लूइड्स हैं उनमें या बायोलॉजिकल सिस्टम्स दूसरे हैं उनमें दूसरे इलेक्ट्रोलाइट्स की कंसंट्रेशन बहुत कम होती है, जिससे कि वो हाइड्रोजन आयन कंसंट्रेशन को बहुत ज़्यादा अफेक्ट नहीं कर पाते हैं. इसलिए हम उनका असर हाइड्रोजन आयन कंसंट्रेशन पे ना लेके पीएच कैलकुलेट करते हैं. It is a general rule of biochemistry that activity for all practical purposes equals concentration, whereas this is not always the case. The rule is almost universally applied. तो बायोकेमिस्ट्री अगेन क्या है? बायोकेमिस्ट्री बायोमोलिक्यूल्स की केमिस्ट्री. बायोमोलिक्यूल्स कहाँ पे कंसिडर किए जा रहे हैं? बायोलॉजिकल सिस्टम्स में. तो बायोमोलिक्यूल्स लाइक कार्बोहाइड्रेट, फैट, प्रोटीन, न्यूक्लिक एसिड, एंजाइम्स एंड प्रोटीन्स ऑफ कोर्स, तो उनकी केमिस्ट्री हम पढ़ते हैं. एंड वो सारे structures and functions are dependent on the pH of the solution in which these are present. So it is very important to know about pH. Now, introduction of pH continues that pH was originally an acronym for French clause pauvre hydrogène, which can be translated into the English as power of hydrogen or potential of hydrogen. Hence, the name is pH. The pH is used to quantify acidic, alkaline or alkaline nature of water-soluble substances, which is measured in terms of hydrogen ion concentration. The pH value is an expression of the ratio of hydrogen ion concentration to hydroxide ion concentration. Hence, if the hydrogen ion concentration is greater than the hydroxide ion concentration in the solution will be acidic, otherwise it will be alkaline or basic. The pH value is a number, it is denoted by a number, and we consider it from 1 to 14, with 7 as the middle or neutral point. Values below 7 indicate acidity, which increases as the number decreases. And that is 7, 6, 5, 4, 3, 2, 1. 1 being the most acidic. Values above 7 indicate basicity. That is 8, 9, 10, 11, 12, 13 and 14. And 14 being most basic. pH meter is an electrical device used to measure hydrogen ion activity, acidity or alkalinity in water-based solutions, indicating its acidity or alkalinity expressed as pH. It provides a value as to how much acidic or alkaline a liquid is. For example, acid dissolves in water and forms positively charged hydrogen ions. The greater this concentration of hydrogen ions, the stronger the acid is. Fundamentally, a pH meter consists of a potentiometer attached to a pH responsive electrode and a reference electrode which is unvarying. pH responsive electrode varies while reference electrode remains constant. The difference in electrical potential relates to the acidity or pH of the solution. pH meters are commercially available to measure pH of the solution. In 1934, Arnold O. Beckman invented the first commercially successful electronic pH meter. This is one picture from UTech company. This is the board or digital screen here, which shows the readings of pH of the solution, and this is the device which measures the solution, pH of the solution. And with the help of these buttons, we adjust various readings or follow the digital display of the pH of the solution. Electrodes of pH meter are two half cells, one indicating pH electrode and the other reference electrode. Most applications today use a combination of electrode with both half cells in one body. You can see this here. These wires are going to pH electrode and reference electrode. Both are contained in one glass or one casing, and both half cells are present inside this, like this. Details of this we will be seeing in next slides. First of all, working principle of pH meter. Why it is important to know the working of the instrument? It is important because several factors can cause the observed value of the solution, observed pH value of the solution differ from the actual value. A pH meter measures the voltage between two electrodes placed in the solution. Potential of one electrode, pH electrode, is dependent, while other remains unchanged, not affected by the hydrogen ion concentration in the solution. The most commonly used pH dependent unit is the glass electrode. The action of this electrode is based upon the fact that certain types of borosilicate glass are permeable to hydrogen ions but not to other cations and anions. Therefore, a thin layer 0.05 to 0.1 millimeter of such glass is interposed between two solutions of different hydrogen ion concentration. Hydrogen ions will move across the glass from the solution of high to that of low hydrogen ion concentration. Since passage of hydrogen ion through the glass adds a positive ion to the solution of low hydrogen ion concentration and leaves behind a negative ion, an electric potential develops across the glass. The magnitude of this potential is given by the following equation. A is equal to 2.303 RT upon F log of hydrogen ion concentration 1 divided by hydrogen ion concentration 2. Now, what are these? E is potential. R is the gas constant. F is Faraday constant. H1 is molar concentration on the inside of the glass. H2 is the molar concentration on the outside of the glass. If hydrogen ion concentration of one solution is fixed, the potential will be proportional to the pH of the other solution. Keep one of these fixed, then the potential or pH, which is expressed in terms of pH, will be proportional to the other solution. Since glass electrode, one half cell contains silver chloride, and 0.1 molar HCl solution, their dissociation constants are as follows. Silver irreversibly dissociates into Ag plus and electron minus, and dissociation constant of this is written or shown by this: Ag molar concentration, electron molar concentration divided by undissociated silver. Another solution is silver chloride, which dissociates into silver and chloride, and dissociation of this will be Ag multiplied by Cl concentration, molar concentration, both are molar concentration of the ions, and finally undissociated AgCl and its molar concentration. This relationship will give rise to dissociation value of dissociation constants of glass electrode. Reference electrode is the other half cell, contains mercury chloride, and saturated solution of potassium chloride. Each of these compounds exists in ionized state, although the extent of ionization may vary widely. Their dissociation constants are given in this figure. So mercury is reversely dissociated into mercury and electron, and dissociation constant will be given by this equation: mercury, molar concentration of mercury multiplied by molar concentration of electron divided by molar concentration of Hg. Hg2Cl2 is twice H2 Hg with positive ion and 2 ions of chloride, and again this is the equation for calculating dissociation constant of Hg2Cl2. Here Hg2 molar concentration of this ion, molar concentration of this ion are multiplied and then divided by Hg2Cl2, which is undissociated. This is the conditions present in glass electrode or pH responsive electrode and the reference electrode. The electromotive force of the complete cell E is given by reference electrode minus glass electrode. Electromotive force is a difference in potential that tends to give rise to an electrical current. E reference is the potential of reference electrode, which at normal temperature is plus 0.250 volts, and E of glass, electromotive force of glass, is the test electrode, which depends on the pH of the test solution. Then the pH of the solution at 25 degrees can be determined by the following equation. At 25 degrees centigrade, substituting the value for E reference, what we will get? pH is equal to E of glass minus 0.250 volts divided by 0.591 volts. And this equation will give pH of the unknown solution. The pH of the solution measures the degree of acidity or alkalinity relative to the ionized water or ionization of water. Measuring pH involves comparing the potential of solutions with unknown hydrogen ion concentration to known reference potential. pH meter converts the voltage ratio between a reference half cell and sensing or pH electrode half cell to pH values. At 25 degrees centigrade, pH 0 is at plus 414.120 millivolts, and this is acidic. pH 4, this is plus 177.480 millivolts, this is also acidic. pH 7, 0.00 millivolts, and this is neutral. Then pH 10, voltage is minus 177.480 millivolts, this is basic. pH 14 is equal to minus 414.120 millivolts, and this is again basic. The voltage on the outer glass surface changes proportionately to changes in hydrogen ion concentration. Picture of glass and reference electrodes of a pH meter. You can see here ordinary glass का बना हुआ है. This is made of ordinary glass. This is silver wire coated with silver chloride. This is this glass, borosilicate part of the glass is filled with 0.1 molar HCl, and here rest of the solution, silver and silver chloride, are filled. Now here this side, reference electrode, this hole is open to air to prevent effects of changes in atmospheric pressure. Reference electrode is also known as calomel electrode. Calomel, Hg2Cl2 saturated KCl. This mixture is known as calomel, and here saturated KCl is filled, and this fiber or capillary is attached here. This is for sensing the difference in change in electromotive forces between the two electrodes. Picture is explained here again. The glass electrode contains 0.1 molar HCl in contact with H permeable glass, which is borosilicate glass. Connection to the potentiometer is by means of silver wire coated with silver chloride, which is immersed in HCl. The circuit is completed by immersing the into the solution a reference electrode that has been selected to be the pH independent. The type most commonly used contains a Hg2Cl2 paste in saturated KCl. This is called calomel electrode. If a high temperature operation is required, AgCl is used instead of Hg2Cl2. In both cases, KCl makes contact between this unit and the solution being measured, KCl. A saturated potassium chloride solution is normally used as salt bridge in the reference electrode because many ions diffuse from salt bridge against which the sample ion must diffuse. So, in the part 2 of pH meter, I am going to explain certain terms which are not explained in this, like salt bridge. So please see that also. Reference electrode is encased in a tube made of glass that is impermeable to hydrogen ions, so that its potential is pH independent. Electrical contact between KCl within the electrode and the solution is by means of fine fiber or capillary in glass casing. The KCl slowly flows into the sample in cases in which chloride ions is not required. Hg sulfate reference electrode can be used. Voltage measured by such a system is primarily the difference between that of glass and the reference electrode. This can be written as follows. Which is V constant is equal to 2.303 RT divided by F and pH. Thus, voltage generated is linearly related to the pH of the solution. This is all for this part. I'm going to explain more things in part 2. Thank you.